Okay, welcome to the electron transport chain with Mr. Party. So we're going to do oxidative phosphorylation, which is the electron transport chain. Let me get the laser pointer up. So the electron transport chain occurs in the cristae of the mitochondria, which are these folds here. The proteins responsible for the electron transport chain can be found on the inner membrane, which is here, and the electrons pass through one complex to another. As this happens, the energy is used from the electrons to pump protons into the intermembrane space, which is this pink thing here, and then the protons then move through the ATP synthase as it does in photosynthesis down a concentration gradient, down the electrochemical gradient, and this is called chemiosmosis. It causes a proton-motive force which rotates ATP synthase, and that causes ADP, beg your pardon, to become phosphorylated to make ATP, which is used for uh, many reactions. So here's a zoomed in diagram of the mitochondria, inner membrane, intermembrane space, and outer membrane. This means this blue box up here is the cytosol or cytoplasm. This down here is the matrix, which is the cytoplasm of the mitochondria. So you've got four. How do I get rid of this? There we go. So you've got four iron containing complexes. All you need to know is complex 1, 3, and 4 exist. And I'll let you go through the rest of it in your own leisure. You also need to know that ATP synthase exists. The rest of it you don't really need to know. So again, you can read this in your own time on the PowerPoint. I'm just going to skim through it. So number one, reduced NADH, which is NADH plus H plus, so you can just write NADH, comes to complex 1. This NADH has come from the Kreb cycle and glycolysis as well. So what happens is when reduced NADH gets to complex 1, it becomes oxidized. That means the electrons are given to complex 1 and the hydrogens are also taken away. And the electron's energy is used to pump hydrogens into the intermembrane space. You don't need to know how many hydrogens, but again, I made this when I was teaching A-level way back when, and they liked it a bit more complicated. They wanted to know the extra information. Okay, so you don't need to know this, but this is a coenzyme called coenzyme Q, and this is just an electron carrier that carries electrons from one place to another. So it's good to know that you should know electron carriers exist, but you just don't need to know the names. So then what happens is complex 3 does the same thing. It's going to use the electron's energy to pump hydrogen ions into the intermembrane space. The only difference between 1 and 3 is that 1 1 oxidizes NADH, whereas 3 doesn't oxidize anything. It just gets reduced and then it becomes oxidized itself when it passes the electrons from one to another. So you don't need to know this part either, but FADH or which comes from the Kreb cycle, also becomes oxidized. You can say this occurs at complex 1 for A level, but there's complex 2 down here. Complex 2 is actually part of the Kreb cycle. So what happens is the electrons get passed to complex 3. There we go. Okie dokie. Now the electrons are at complex 3, then cytochrome C, which is another electron carrier, is going to pass the electrons from complex 3 to complex 4. And if you want to know, cytochrome C can only carry electrons one at a time, whereas a coenzyme Q can carry them twice, two at a time. So yeah, and complex 4 does the same thing. It's going to pump hydrogen ions into the intermembrane space. And now if you notice, we'll get to this in a bit, there's a high concentration of hydrogen ions in the intermembrane space. The last thing now is that these electrons can't stay here. Now they've lost all their energy, that's why. So at complex 1, the energy was used to pump hydrogen ions. Complex 3, electrons energy is used to pump hydrogen ions. And then complex 4, electrons energy again was used to pump hydrogen ions into the intermembrane space. This means something has to accept the electrons, and the final electron acceptor is oxygen. So there we go. So oxygen becomes reduced because reduction is gain. Complex 4 becomes oxidized because oxidation is loss of electrons. So some quick notes, you can look through these yourself later. Let's get back to the nitty gritty, which is chemiosmosis. So you've got a high concentration of hydrogen ions here. This means you've made there's a high concentration gradient between the intermembrane space and the matrix. There's also a high electrochemical gradient between the intermembrane space and the matrix. These gradients also exist between the intermembrane space and the cytoplasm up here, but we're mostly looking at here. Now, the hydrogen ions aren't able to move either way. They're trapped in this space, and there's only one channel that lets them go through, and that's ATP synthase. So what happens is the hydrogen ions move down these gradients, electrochemical and the concentration gradient, and as a result of this, it causes something called the proton-motive force, which is this turning movement of this bottom subunit of ATP synthase, and this causes ADP, adenosine diphosphate, notice it had two phosphates, to become ATP, adenosine triphosphate. So watch this one last time. Okay, and this P, the lonesome P you see here, that's an inorganic phosphate molecule. So inorganic phosphate plus ADP becomes ATP. This process of hydrogen ions moving down a concentration gradient is known as chemiosmosis. Okay, and that's pretty much it. Here's a nice song that you can look into. I really like it. It shows everything really well. The guy's proper into his music there. Here is a simplified process. So NADH becomes oxidized, hydrogen is removed, electrons' energy is used to pump hydrogens into the intermembrane space by complex 1. The electron is then moved to complex 2. The energy is then again used, electrons' energy is used to pump hydrogens into the intermembrane space. The electron is then moved to complex 4. The electron's energy again is used to pump hydrogens into the intermembrane space. And then finally, the electron is accepted by oxygen because the electron's so low in energy. At complex 2, and you can say this occurs at complex 1 as well, FADH loses an electron, so oxidized, FADH becomes oxidized, and the electron does the same thing. And FAD and NAD plus are then recycled back to glycolysis, Kreb cycle. High hydrogen ion concentration causes an electrochemical and a concentration gradient to build up, which causes hydrogen ions to move down these gradients, which causes a proton-motive force, which turns the bottom subunit of ATP, which phosphorylates ADP into ATP. And that's pretty much the whole thing simplified. This is a Gibbs free energy chart, which just shows the electron's energy reducing every time a proton is pumped by complex 1, 3, and 4. Notice at complex 2, the energy is not lost by the electron. It stays the same until it reaches complex 3. So this is what's driving the hydrogen pumping. So there's some more diagrams throughout you can look. Here's an interesting question I thought we can look through right now. A new discovery found that protein structure of complex 1, 2, 3, and 4 in mitochondria of cancer cells are slightly different from other cells. Discuss how this could be used to cure cancer. So you could say that they could design an inhibitor that's specific for these proteins, which will inhibit their action, which will then stop the respiration in cancerous cells. Something along those lines will get you the marks. So there's for more information, the main thing is that there's 28 molecules of ATP produced during the electron transport chain. This means with the addition of the 2 ATP that are produced in glycolysis and 2 ATP that are produced in the Kreb cycle, that's a total of 32 ATP produced in aerobic respiration compared to just 2 ATP molecules produced in anaerobic respiration. Okay, so I'd say finish this off, and that's pretty much it.